Apologies, everyone, for delay in starting. We've had some technical glitches at this end, but we will begin with your questions now. I will first turn it over to Jam to welcome Dr. Kang.
Yeah, thank you, Ritika. Firstly, again, apologies for the delay, and it's a pity that we can't see Dr. Kang. It's a pleasure to welcome Dr. Gagandeep Kang to the ISRC webinars. She's the Executive Director of the Translational Health Science and Technology Institute, Faridabad, and one of the noted clinician scientists of the country. She's a medical scientist who has worked on diarrheal diseases and public health in India for a very long time, a key contributor to rotavirus epidemiology and vaccinology in India. And she's done extensive work on vaccines and explorations of different approaches to improve the performance of oral vaccines. And her work has led her to being described as India's vaccine godmother. So it's a pleasure to welcome India's vaccine godmother, Dr. Kang. Thank you. We look forward to the interaction with you.
Thank you, Jam, and thank you again, Dr. Kang, for joining us. We are going to start with the questions that we received from registered participants during the registration process, and during the call, we will also be able to take your questions. Please type them in during the Zoom chat, and we'll take them as we go along. Thank you to everyone who's joining us from Facebook Live. Please send us your questions over there, and we will do our best to incorporate them in the program today. So to begin with, Dr. Kang, could you tell us briefly about the steps in the process of vaccine production, and specifically in terms of COVID-19 vaccine, what are currently the hurdles?
So if you want to make a vaccine, you have to start with choosing your antigen. And it seems pretty clear that for SARS coronavirus 2, most people are going for the whole or parts of the spike protein of the virus. This is not unusual, because if you want to look at what is likely to induce an immune response, it tends to be proteins that are on the surface of viruses or bacteria that matter most for immune responses. So once you've identified what your immunogen or antigen is going to be, you have to look at what your delivery system is likely to be. You might decide to deliver the antigen as part of the whole virus, and it could be live virus or inactivated virus. It could be just the component, so make it into a subunit vaccine, and you could also choose to deliver it in many different ways. You could send it as a message, which then asks the host cells to make the protein immunogens. Those are what DNA and RNA vaccines are based on. So there are multiple approaches to choosing how you will deliver an antigen. In order to see whether this works or not, usually we would try to step up the delivery systems, going initially from testing these in animal models in preclinical studies, and then move to testing in humans. Usually the first step in humans is to test for safety, then for immunogenicity, and then finally to see whether the vaccine really works to prevent disease or not. Depending on the approach that you've taken, you might want to add other things to the vaccine, such as adjuvants, which could enhance the immune response. So there are a lot of steps that are required for making vaccines, but it's very important to remember that the first thing about vaccines is that they need to be both safe and efficacious. Because if safety is a concern, that vaccine will not become a viable product. Vaccines are given to healthy people, and you don't want to make them sick by giving them a vaccine that may be unsafe in some way.
So when vaccines have to be fast-tracked, like there is a push right now for COVID-19, what are the steps from the usual protocol that are different?
If we look at what's been happening with SARS-CoV-2, there's been an unprecedented effort to move vaccines quickly. But in that movement, neither safety nor efficacy can be compromised. So if we take the example of the first vaccines that went into humans, what was done was to rely on data for platforms that had used those technologies before for safety. So for example, if you have an mRNA vaccine, it is only the message that is different, the sequence of the mRNA. So if mRNA vaccines made with that platform have been used for delivering many other antigens before, can you rely on that safety data to decide whether or not you can get into humans? This is what was done by the FDA for the Moderna vaccine, and similarly for the Inovio DNA vaccine. You can also do things in parallel. You can try and accelerate studies by doing safety and immunogenicity together rather than sequentially. In some cases where the vectors or platforms are really well known, regulators have allowed manufacturers and developers to go ahead with doing animal and human studies in parallel. So there is going to be no shortcuts to testing whether vaccines are safe, immunogenic, and effective. The only thing that might change is that if there is reasonable proof that the vaccine is likely to work, if we can by that time have a correlate of protection, then we may be able to license the vaccine without necessarily waiting for clinical efficacy and gather the data on the efficacy of the vaccine post-licensure. This has been done for other studies before. For example, for pneumococcal vaccines, we do license based on a presumed correlate of protection, which is antibodies above a certain level. So we are not going to be compromising on getting these vaccines licensed. The testing pathways have to be the same as are used for all other vaccines. The only thing that will change is to allow for faster process and parallel process instead of sequential.
So with that, the question on everybody's mind is, when can we expect a vaccine for this?
If we are really lucky, and if Donald Trump has his way with warp speed, we will have vaccine available somewhere in the world by the end of the year. I actually think a more likely timeline is sometime middle of next year. It's important to remember that having a vaccine doesn't mean that the whole world will be vaccinated. A lot of the vaccines that are very fast to make, like the mRNA and DNA vaccines, where we were able to have the vaccine into humans within less than 3 months of the virus sequence being described, are also ones where making them at large scale is very, very challenging. It's easier to make protein vaccines than it is to make DNA and RNA vaccines. So having a successful vaccine will not necessarily mean that we will have enough vaccine to vaccinate everybody, even if we do get a successful vaccine next year. Now, if you look at how many projects there are over 100 projects that are looking at making vaccines around the world. Most of these projects are going to fail. They will fail at different stages, but it's very unlikely that we are going to have 100 successful vaccines. If we get 2, 3, or 4, that would be really great.
So the we've heard that there are some of the vaccine candidates are already in human trials. So what is the new information that we have learned from those?
The vaccine candidates there are 8 vaccine candidates in human trials. And as I said, the first thing that you learn about a vaccine is its safety profile, and safety is usually done in a small number of participants. Only 2 vaccines are moving from phase 1 to phase 2 currently. And these are a vaccine made in China and a vaccine made in the US. So what we know from those 2 are that so far there are no safety signals that would lead to the termination of those trials. So with each vaccine that comes along, we are going to have to first look at safety and then look at immunogenicity. So in terms of human trials, the only thing we have learned so far is what are the safety signals. 2 of the vaccines have been put into primates, and in primates, they were immunized with the vaccine that is being developed out of Oxford and a vaccine that is being developed in China. And in the US, the Oxford vaccine went into 6 monkeys, and China, their vaccine went into 8 monkeys that were then subsequently challenged with wild type virus, and the monkeys did not develop severe disease. So the only proof that we have, or only suggestion that we have, that the vaccines might induce an immune response, a good immune response that may be protective, really come from these primate studies. But the primate model is not a perfect model for the kind of disease that is seen in humans.
So what does that mean, that there will be subsequent steps that will have to be required between primate studies and finally going for the same in humans?
The primate studies are done to complement the data that is gathered in humans. And there is no question of primate studies being a replacement. That can happen. There is something called the animal rule, which is applied by the FDA, where if you have a really good animal model of a disease and you can show that there is protection in that animal model, you can apply under the animal rule for licensure of the vaccine. But what we know is that primates are not a particularly great model for SARS-CoV-2 infections. Therefore, it's unlikely that we will be able to use the animal rule as a way of licensure. So the only option we have is to generate data in humans. And this will have to follow the phase 1, phase 2, phase 3 route. The only possible sidestep might come if we have a rescue treatment that is discovered, or if remdesivir works really well. And we could potentially then consider doing human challenge studies that would give you data on whether the vaccine works in humans or not. Usually when human challenge is done, it's considered a late phase 2B study. It's not considered equivalent to a full phase 3 trial. But if we do get to the point of human challenge, that might speed up our ability to develop new vaccines and could lead to us discarding a lot of candidates that are likely to fail in phase 3.
Okay, so it's more a parallel process from what we learn from animal studies and as it feeds into the human trials. Is that correct?
I think for any vaccine, you want to try and generate as much data as possible, both from animals and from humans in the largest possible numbers, so that you are reasonably sure that you have reliable results. that can be quite challenging to do, especially in a short time frame, and if you have many competing candidates. If you are to think about it, look at what's happening with the pandemic now. Different places are being affected at different times. So if you want to get in somewhere and do a phase 3 efficacy trial, you have to carefully time the studies. You almost have to predict where there's going to be a lot of disease, get in there before the disease arrives, immunize everybody, and then see whether they develop disease or not. So there are lots of logistic challenges to evaluation of vaccines in the context of a pandemic, but these are things that are going to need to be done.
So then when we say, let's say, in a year, if we have a vaccine, then we're talking before these phases in humans that you've described.
Yes, we are going to have to do all these phases in the next 1 year if we want a vaccine by next year. And many of the plans that have been developed for the leading vaccine candidates do have that kind of time frame. The problem is going to be, as I said, for phase 3, when you want to produce, efficacy data, you are going to need to be in a place where the disease is going to arrive. So that can be quite challenging to do, but these are things that we are planning now. What would a good field site look like? How many do you need to have? If you prepare 20 field sites, will you be able to use 5, 6, or 10 of them? We don't have answers to all of that yet for phase 3 trials. Phase 1 and phase 2 are all right because those don't require people to get the disease. Phase 1 is a safety study, so you're not measuring a clinical outcome. Phase 2 is immunogenicity and dose finding, and again, you're measuring an immune response and not a clinical outcome. So these can be done anywhere where vaccine is being developed. But if you want to evaluate efficacy, you have to be in a place where there is going to be disease. And that's a challenge.
So the information that's going to be needed to arrive at those decisions, is that something that can be discussed now, or we have to wait for results from previous steps to get there?
So one of the things obviously we'll need the safety and immunogenicity data before we move to phase 3. But one of the things that we can do to prepare sites is look at site readiness. And one of the issues with site readiness is you don't want to give the vaccine to people who have been previously exposed to the virus, because they will already have made an immune response and be protected. So when you identify a site, you have to identify a site where both not too many of the people that you are likely to recruit have already been infected, plus one where the disease is likely to come back. So this is a bit complicated to do, but is one of the approaches that we are thinking about for choosing the right kinds of sites. And as I said, we might start out establishing many, many different sites and then only be able to use a few, because that balance of prior exposure and future disease needs to be met.
Okay, so we've gotten a lot of questions on the general immune response and how vaccines sort of facilitate that. So on the one hand, a lot of questions seem to be around the idea that when people fall sick, it's because their immune system isn't working as well as it should. The pathogen has the upper hand. So then people should either eat this or drink that, vitamin C, what have you, to get the immune system to working to the capacity that it should. So is it true that when people fall sick of anything, it's because the immune system is not working as well? And is there a linear relation to what we eat?
So our immune systems are fantastic. They are just phenomenal in the way we recognize pathogens and react to them. And the immune system is one of the things that is most protected. So people talk a lot about malnutrition and what it might do to the immune response when children are vaccinated, for example. There are a few situations where lack of appropriate nutrition does affect the immune response. And the ones that we do know something about are zinc, vitamin A, and vitamin D. Now, almost nobody in the world, or most people in the world, are no longer vitamin A deficient because we've had supplementation for quite a while and we get it from food. Zinc and vitamin D deficiencies are relatively common in places like India, but those seem to be responsible or affected for certain kinds of infection. So zinc, for example, we know is important for some bacterial and some viral respiratory and gut infections. And vitamin D, the jury is still out, but it's been shown to have a role by some people for some infectious diseases and not from others. So if we think about what supplements you should be taking, I actually think that you really don't need to take anything. Most people who are reasonably well nourished will not benefit from adding more vitamins or more of certain kinds of foods. If you think it's going to be of value, certainly go ahead and take whatever makes you comfortable. But in terms of evidence-backed information on what dietary supplements might help you against SARS coronavirus 2, we really don't have anything at the moment.
Okay, before we go ahead, just a quick reminder to everyone, please type your questions in the chat box here. We will take them during the call, and also from the same for Facebook Live. So one question we have from the audience right now is the connection between coronavirus and in pets, in cats and dogs, and can people contract them from their pets?
So it goes more the other way around. So the evidence that we have at the moment is that humans who are infected have transmitted the virus to their dogs and cats. Cats seem to be more susceptible than dogs. And there is an experimental study that has just come out that has demonstrated that cats can transmit to each other. But so far, it doesn't appear likely that we will acquire infection from our pets. We are more likely to give it to them. The virus does replicate in animals, but so far there have been no cases of transmission back to another human.
Another question from participants right now is if immunity against other coronaviruses protect against COVID-19?
So based on cross-reactivity studies, it is feasible that people who have antibodies to the spike protein of SARS coronavirus might have some level of protection against SARS coronavirus 2. For seasonal coronaviruses, the spikes are sufficiently different that there does not appear to be cross-reactivity for the spike protein, and it's unlikely that there will be protection. It's important to remember that there are only about 5,000 people in the world who have had a SARS coronavirus infection and are around. So therefore, let's treat it as you're not going to be protected from having encountered another coronavirus.
Another question we're getting is the effects of nicotine on the infection. There are many articles coming up on that right now.
Yeah, so I think the most important thing, well, stop smoking. That's the first thing. That's good for you, whether there's SARS coronavirus 2 around or not. The question is one really of lung damage. And we know that what nicotine does is alter the movement of your cilia so that they are unable to clear any infections. So while the data are still emerging, we know that anybody who has compromised lungs does worse with a SARS-CoV-2 infection than people who have normal lung function.
Okay. We also have a question on alternative therapies apart from vaccine. What about stem cells or the CRISPR technology? Do they have potential for treatment?
I think you need to look at, okay, my answer to that is get real. So far, if you have a virus that is going to infect everybody in the world at some point in time, what is the likelihood that technologies that are based on stem cells or CRISPR are going to be scalable? Who will they be available for if they do become available? We can't afford to treat people for cancers. Are we really going to be able to use these technologies to treat people for an infection? Even if the treatments do work, I think these are going to be available for a very, very tiny fraction of the population. So what we should be looking for is cheap, scalable therapies. And that's what the trials that are ongoing at the moment. I mean, what was the promise of hydroxychloroquine? It was really around, here's an available drug that might just work. And if it does, we can give it to everybody. It does seem like that promise is not going to hold up as we had hoped, but there might be others as well that we could think of trying out. Many other available drugs have been proposed, and I think the chances of coming up with something that will function as a public health tool is better with available drugs or later with new chemical entities than it is with new biological therapies, because as we know from all the MAB studies that have been done, all the vectored therapies that have been developed, that these are very expensive and are therefore limited to very, very tiny numbers.
Another question we've got from viewers now is on the fact that people who succumb to COVID-19 infection, the reason for their death is excessive immune function. So how does a vaccine change that? Or how, in sense of how does the fact that excessive immune function is the reason for patient death, how does that feature in vaccine design strategy?
So the vaccine is given before you develop infection. So you're not going to have viral replication at a level that will lead to immune dysregulation. At least that is the hope that we have when we make vaccines. We want the vaccine to induce an immune response that is protective. It's likely that that immune response is going to be largely antibody-based. And if you make that immune response, you will be able to suppress virus replication to a level where you will not wind up in the kind of situation that comes from progression of disease. So, sorry. One thing that I will add is that the only thing that we have to worry about with vaccines, and something that has been brought up with by a lot of people, is that you don't want vaccination to make subsequent SARS coronavirus 2 infection more severe. Now, this has happened before when we've had vaccines that have been based on whole inactivated virus with an alum adjuvant, and also with the respiratory syncytial virus vaccines in children in the 1950s. Now, one of these, of course, is known as antibody-dependent enhancement. This is something that we know from dengue viruses and dengue vaccines most lately. So ADE is basically an FC-mediated enhancement of the infection that's associated with dengue viruses. And this can now, we have tools to measure this. It can be measured in vitro on cells that express the FC receptor, either naturally or by transfection. And this mechanism essentially involves better binding of the virus antibody complex to FCR-bearing cells. And that triggers viral entry, so you wind up with a lot more virus getting in and producing severe disease. So this happens when the vaccine-induced antibody is insufficient to neutralize the virus. So a sub-neutralizing level of antibodies can lead to antibody-dependent enhancement. So this is something that we know about and we'd like to measure when we develop vaccines. This has been shown to occur for SARS coronavirus 1 in an in vitro experiment where FCR-bearing human B-cell lymphoma cell lines were used. However, we don't know whether what we saw in vitro is actually going to show up in an in vivo approach to vaccination. So that's one way. ADE is one way where a vaccine could wind up being unsafe if it produces sub-neutralizing levels of antibodies. There is another safety concern, and this is something called vaccine-associated enhanced respiratory disease. And this is a clinical syndrome that was first described for respiratory syncytial virus when whole virus inactivated vaccines were developed. When these vaccines were given to children, it resulted in a more severe disease in the children. And that was because the antibody that children were producing was not a neutralizing antibody. So they had a lot of antibody that bound, but could not neutralize the virus. And in the presence of a high viral load, this resulted in immune complex deposition and complement activation. So this then, some children actually died, and an RSV vaccination program was stopped for almost 50 years. After that, we found that the reason why this was happening was because it is the pre-fusion F protein in RSV that matters. And the antibodies that were being made were actually being made to the post-fusion F protein. So these were two situations where vaccines enhanced disease instead of preventing disease. And this is something that we need to make sure that we monitor as we focus on developing vaccines for SARS-CoV-2.
This next question from our audience here at Zoom is, currently, what are the best ways to treat patients? Is remdesivir our best bet?
So the best way to treat patients right now, we know for sure that supportive therapies work. The bulk of patients do well. Well, you have to remember that there is a large component that is asymptomatic infections, a large component that is mild infections, a large component that is moderate infections, and a small component that is severe infections. The severe infections are what land you in hospital. And we know that the first sign of a severe infection is a drop in oxygen saturation. And this is being referred to as happy hypoxia, because people don't know that their oxygen saturation levels are dropping. And when they wind up in hospital, by the time you are short of breath, your oxygen saturation is really very low. So the first thing that needs to be done is to bring that up. And the best way to do that is by giving supplementary oxygen through a nasal cannula, and to go as high as you can on that oxygen. These are high flow nasal cannulas that can be used. We have come to understand a few things about SARS-CoV-2. One is that you want to keep people off ventilators for as long as possible. In the initial days of the pandemic, it was thought that we should get people onto ventilators as quickly as possible. But it's been found that people who go onto ventilators do quite badly. So now the focus has shifted to making sure that people stay off ventilators. And one of the new things that we've learned is that a technique that is called proning, which is very rarely done for people who are ventilated because it involves turning the patient around, flipping them on their stomachs. This works quite well with the nasal cannulation, and it leads to better aeration of the lung. So we are learning as we go on what needs to be done for respiratory support. The other things that we are learning is that this really affects the blood vessels and multiple organs. So you can have the brain being affected, you can have the kidneys being damaged, people winding up needing dialysis, and you can have thromboembolic phenomena. And these will require anticoagulation. There is discussion around what is the right timing for administration of steroids. So in terms of management of SARS-CoV-2, even before we get to unproven or promising sounding antivirals, there is a lot that can be done. Remdesivir is one antiviral that we've been told shortens the duration of illness by about 30%. So that's good news, but it's not a miracle or wonder drug. So then the questions that need to be asked is, when should you be giving it? Who are the patients that will benefit the most? Will it work if you give it after a person is severely ill, or should you be trying to get it in as early as possible? Remember, remdesivir is given as an IV infusion. So you have to be in a hospital and getting that IV for 10 days. Gilead is now trying trials where they're trying to shorten that treatment to 5 days. So if it works, then there will be more doses of remdesivir available. But right now it looks like there's only about enough for 150,000. So this is not a miracle we will have tomorrow. It's promising, but we have a long way to go with proving how well it works, when it should be used, who it should be used in. Similarly, with the other antivirals that are in trials, we have some evidence that some have failed in trials, and we will keep trying till we get an effective treatment.
Of the different effects that you mentioned, effects on blood vessels, people on the brain, for example, to what extent are these effects directly because of the virus or a body's response to the virus?
By the time you get to really severe disease, which tends to happen around 9 or 10 days, most of the effects that you're seeing are host response to the infection. It's not directly virus infected. What does the virus do? It gets inside cells, it multiplies, it comes out of the cell and goes and infects other cells. So the virus, in terms of replication, will continue to make more virus till the immune system kicks in and tamps it down. Now that happens usually by the time you're making an antibody response, which is 2 weeks after you've gotten an infection, you've got reasonable antibodies. So most of the severe disease that we are seeing is really coming from host response. Remember that the antibody response is very effective. This is why people have asymptomatic disease, mild and moderate disease in the majority. The severe end of the spectrum is a small component, and most of that is the manifestations that we see are a consequence of host response.
So in that case, you're saying the people who are the most sick, it's a function of their own immune response that makes them so much sicker than others?
To a large extent, yes, that is the case. So it's not just about the immune response alone. As I mentioned, there are thromboembolic phenomena, which could be more inflammation if you separate inflammation and adaptive immunity. So yes, it is largely an immune response that is immune response and inflammation that are responsible for this.
So in this context, there is a lot of talk of immune boosting, to take this or that amla or chyawanprash or whatever to boost your immune response towards this. Then could that backfire in this case?
I doubt that will backfire. I think the idea needs to be evaluated in clinical trials. Any treatment needs to be rigorously tested. And the problem with things that are done as a prophylactic is that sometimes that can be very hard to measure in either biology or in the kinds of contexts that we do testing in clinical settings, which is a randomized controlled trial. Not to say that it can't be done, but those are studies that would need to be very carefully done. And I'm not sure that anyone is prioritizing those kinds of studies at the moment.
Another question that is coming up now, and we've gotten a lot earlier also, is that how confident can we be in terms of developing a vaccine for COVID-19 when other diseases like AIDS and malaria that we've known have been around for a long time and there are no vaccines for those?
Okay, so AIDS and malaria have been around for a long time, but the AIDS virus mutates very rapidly, right? That's the reason why we haven't been able to get a vaccine so far. And now we are looking at whether we could have the limited number of broadly neutralizing antibodies that have been identified from patients as a strategy to prevent infections with HIV. It's something that has promise. So if you could have monoclonal antibodies that you know are broadly neutralizing, could you put them into a microbicidal cream? Could you think about doing post-exposure infusions? So still looking at that same principle of antibodies protecting you against disease. For malaria, we do have a vaccine that is about 30% protective. But remember that malaria is a pathogen that has multiple stages within the human host. So finding a single thing to target and then looking at whether you can shut down disease or shut down transmission or shut down infection has very different connotations for a multi-stage pathogen. than it does for something like a virus, where all viruses are pretty much the same. If you look at vaccines that have been successfully developed so far, most of the vaccines that we have are viral vaccines. It's way easier to make a viral vaccine than it is to make a bacterial vaccine. And bacterial vaccines are easier than parasitic vaccines, where essentially we only have the malaria vaccine at the moment. So I'm hopeful that given the tools and technologies we have today, we should be able to come up with a successful vaccine for SARS-CoV-2. We came pretty close for SARS, and because the disease died out, there was no possibility of taking the testing of those vaccine candidates further. So a lot of the lessons that we learned from SARS are now being applied to SARS-CoV-2 with much better technologies. And that's one reason why we have been able to move this far, this quickly.
In people who are immune suppressed, who have other chronic conditions, who are also said to be at most risk for contracting COVID-19, what precautions would you say that they should bear?
I think for everybody who is at increased risk of acquiring the infection, and this includes not just those who are immunocompromised, but the elderly and people who have comorbidities, we should be looking at the best ways to protect them from infection. There is no treatment at the moment. So what we need to do is to ensure that until there is a vaccine, and a good vaccine, that we try and make sure that they don't get infected. It may not always be possible 100% of the time, but we should do our best to ensure that they don't get infected. It's important to remember that there are many viral infections that can be handled reasonably by people who have some level of immunocompromise. A lot depends on what kind of immunocompromise it is. So best case scenario, protect from infections, but if they do acquire infections, that does not mean automatically that they are going to get really, really sick with this virus. It may happen, but it's not guaranteed.
Another question from our audience now is, say we develop a vaccine, or someone, or more realistically, somebody recovers from COVID-19 on their own. The immunity that they develop as a result of it, how long will that give them protection afterwards?
So if you develop an infection, you do make antibodies, and what we measure with these serodiagnostics or serology tests are really the presence of binding antibodies that have been developed in response to an infection. The protection comes usually from most viruses from neutralizing antibodies. And there is a difference in reports that have come from different groups about both the proportion and the quantity of neutralizing antibodies that are made by people who are infected. There are several groups that argue that if you go out at 28 days post-infection and measure, practically everybody who has made an IgG response will have neutralizing antibodies. But recently there have been a few papers that say that neutralizing antibodies might be very low in some people. Now, how much neutralizing antibodies matter, we don't know yet. We know that, for example, in other viruses like polio, when you make a neutralizing antibody response, the range of the response is very large. But in order to have protection from polio, you need a fairly small amount of neutralizing antibodies at a titer of 1 in 8. So what the cutoff will be for SARS-CoV-2 for neutralizing antibodies, if that is a correlate of protection, we don't really know. How long will these last? Well, we have some information from MERS and from SARS coronavirus. SARS coronavirus neutralizing antibodies lasted about 2 years in the bulk of people who had an infection. MERS coronavirus antibodies were slightly less time, about a year. But even when you no longer have measurable neutralizing antibodies, it's possible that you might have made good memory. And then when you get infected again, you may make antibody quicker. So we don't know yet. And this is something where tracking patient cohorts of people who have been infected previously to see how long their neutralizing antibodies last and how much of the antibody they make is going to be very important for our understanding of disease. These studies are ongoing in many parts of the world.
Could you tell us about the WHO Solidarity trial?
Sure. So WHO actually has 3 solidarity studies. The first one is the drug trial. And this is comparing outcomes that really matter. Did a person get discharged or die? Across a range of different interventions. The interventions that are being tried in Solidarity right now are looking at lopinavir, ritonavir with and without interferon 1 beta, hydroxychloroquine and remdesivir. There are discussions about adding additional arms for new interventions. The nice thing about Solidarity is it's being done as a study across multiple countries. And once you recruit your patient into the trial, you have to fill out a lot of information online, but then the outcomes are these very specific outcomes that are easy to enter. So the trial's been designed so that very busy clinicians can enroll their patients into the trial. The goal for Solidarity is to reach about 10,000 participants by sometime in June. And the last I checked, they were at a few thousand.
So not enough enrollment, you're saying?
No, so Solidarity only got started about 6 weeks ago. And in order to enroll patients into a trial, you have to have protocols in place. You have to train the investigators. And much of this training is happening online with WHO. But for example, from India, I think at the end of last month, we had one hospital that had recruited 1 patient into the trial. So there is a bit of a way to go, but the idea is that by June, we should have something to look at.
Okay. So we're getting actually close to our closing time. So one last question. If you had to recommend 3 things to people for most effective ways to prevent COVID, what would they be?
Wear a mask, wash your hands, stay away from people. By now, everybody should know this. And I think people forget just how effective these methods can be. The last thing that I will say is that if you do happen to get infected, don't blame yourself and don't blame anybody else, because this is a very infectious virus. And many of the people who will spread the virus to others don't know that they have it. So it's very important that you recognize that despite our best efforts, we may still get infected. And if we do, we deal with it and move on.
Thank you, Dr. Kang, for taking our questions. To our viewers, both here and on Facebook Live, I would say that if we weren't able to take your question today, our next webinar will also be about immunology. So please do send us your questions and do join us next time. I will now turn it over to Jam to close the session. Jam?
Okay, yeah, thank you very much. Thanks, Dr. Kang, for the interaction. It's been quite illuminating. I think people have more doubts and these discussions have to go on. I would also like to tell everybody about ISRC, the Indian Scientist Response to COVID-19. Ritika, can I do a share screen and show the ISRC site for a minute? Yeah. Oh, okay. No, I'm not able to do that. But I invite everyone to visit the site. Oh, yeah, I can. Well, I am able to start the video, but I'm not able to share screen. I invite everyone to visit the site IndSciCOVID.in. We have a number of resources here for the public and for resources on mathematical modeling, on apps, on hardware, many things. Several of our busting hoaxes sites, question answers, and daily life COVID series, which also available in voiceovers with professional storytellers giving us stories. So I invite everyone to take a look and visit the site and make use of the resources. So thank you all for coming and joining us today. Thanks again, Dr. Kang, for a wonderful interaction with all of us. Thank you.
Thank you for having me. Bye.
Okay.
Thank you.
Bye-bye.